forgot the name.
Good.
Sorry, I'm gonna find my notes. Oh, so at like a part of it, it like explains that all of the like enzymes and proteins are like on the outside of a membrane, and I just like don't really like understand like how like each one like works so well. Like if you could like explain that, like how the protein.
Most of them, in fact, are in prokaryotes. Most of them are either embedded in the membrane or on the inside of the membrane. Especially those associated with metabolic pathways. Some of them, others may occur elsewhere as well. But if you just have a look at the structure of the membrane, for example, we haven't got there yet, but in a respiratory pathway, for instance, all of the enzymes would be linked here on the membrane here.
Okay.
So when in prokaryotes, when proteins are associated with a membrane, they are only associated with the cell membrane because that's the only membrane that there is. In eukaryotes, the proteins are maybe associated with the membranes of organelles. So, for example, in the eukaryotes, the respiratory enzymes are not associated with the cell membrane. They're associated with the membranes of the mitochondrion.
Okay. Do we have to understand like how each one each like structure of like of them, or
no?
Or,
you mean like what I've just mentioned, the respiratory ones? No. What you do need to know about is the difference, for example, between channel proteins, which are embedded in the membrane, which are a simple channel for specific ions, and the carrier proteins, which actually change shape in order to transport, and then the carrier proteins which are responsible for active transport.
Okay.
And those are these which actually use ATP in order to pump things against their concentration gradient.
Okay, thank you.
The only other thing that you will need to know about, sorry to flip through like this, I know it's confusing, but I want to find the other thing that you'll need to know about enzymes is how their structure is composed of usually very, very long polypeptides, which are then wound into their secondary structure and then into this tertiary structure in three-dimensional structure in space to create an active site where the actual catalysis takes place. That's where the reaction that the enzyme is responsible for is going to take place. It takes place here in the active site, but remember that it may have some enzymes have another site, which is the allosteric site, which is a control site, which can switch the enzyme on and off, depending on what binds at the allosteric site. Okay, so that's basically all that you need to remember about the structure of the enzymes. Apart from the fact that enzymes may also require something else to bind to them, which is a cofactor, sometimes a metal ion, sometimes an organic compound. And organic cofactors are called coenzymes, and they bind, or they can bind anywhere. It depends on the enzyme. So the protein portion of the enzyme on its own is called the apoenzyme. When it's bound to everything it needs and it's ready to go, ready to function, it's called the holoenzyme.
Okay.
All right.
All right. Thank you.
Sure. Anything else? Any others that you can think of for the moment?
Not really, no.
Okay, all right. See, as to other people have joined, has anybody else got anything to ask? Feel free to do so.
Hey, Professor.
Yes, go ahead, Isabella.
Sorry. So about this allosteric sites,
Correct.
they're used to help enzymes and also like make they don't work and make they work, like.
Yes, let's go. Let me find a slide.
Okay, thank you.
Where am I? Here you are. Okay, so first of all, just so that we get it absolutely clear, okay, the enzymes very often require what I've just mentioned. They very often require some other substance to bind to them in order to work. That we call the cofactor. That has nothing to do with allosterism at all. That is part of the functioning of the. So what I'm going to talk about now, all the binding and everything I'm talking about now has nothing to do with the cofactors, all right? The issue of allosterism usually arises when we're talking about a metabolic pathway. In a metabolic pathway, we have a series of enzymes, each of which produces an intermediate, and they end up producing an end product of some sort. The control of that pathway is often because the very first enzyme is an allosteric enzyme. So in this diagram here, here's the active site there. And what this enzyme is going to do, it's going to catalyze a reaction where threonine here is going to end up being turned into isoleucine there. That's the end product. So that's the whole point of this whole pathway. Take threonine and turn it into isoleucine. Like the very first enzyme here it's called Threonine Deaminase. It is an allosteric enzyme. There's its allosteric site there. Okay. And this allosteric site actually binds isoleucine. Well, this may sound strange because isoleucine is the end product here. And when I bind isoleucine here, I'm going to slow down the activity of the enzyme. The way in which this works is this binding of isoleucine is not a permanent binding. It comes and it goes, comes and goes, comes and it goes. When it binds here, it actually changes, it binds here, it changes the shape of the active site. And the active site can't bind threonine anymore. So what is happening is the more isoleucine I have here, the more time this enzyme will spend unable to break down threonine. The less isoleucine I have, the more time this active site has available to break down threonine and start the pathway. So this is a very neat way for this pathway to be controlled by the concentration of its end product. We've got the cell needs isoleucine for something. It is using isoleucine up the whole time. So the cell has to make isoleucine. It does so by taking threonine, pushing it through this pathway, and producing isoleucine. But the cell only needs just enough isoleucine. It doesn't need more isoleucine. And more isoleucine than it can use is a waste. So what is happening is this isoleucine, the product here, is the allosteric inhibitor here. So if the concentration of isoleucine rises, it shuts down the enzyme, and then the concentration of isoleucine begins to fall. when the concentration of isoleucine begins to fall low enough, then the enzyme operates more frequently and makes more isoleucine. It means that the cell has fine control over the concentration of the end product at the end. So this is called feedback inhibition, because what is happening, the end product is feeding back to the start, and it's inhibiting the enzyme. There are systems, they're not as common, but there are systems which are feed forward. They are allosteric, and they are run by an allosteric stimulator. And in that case, what would happen? There is not the case in this particular pathway, but there are some systems where the presence of the substrate switches the enzyme on. So in that case, the enzyme stays, it can't work. It just stays silent until the substrate appears. And as soon as the substrate appears, the substrate is actually able to bind at the allosteric site, switch the enzyme on, and push itself through the path, push through the pathway. So that is a feed forward. Feed forward is switched on by the substrate. Feedback is switched off by the product. That's the simplest possible explanation.
Okay, thank you so much.
Okay.
Yeah, helped a lot.
Let me get back here. Anyone else?
And just one more question.
Go
Do
ahead.
we have to know about like the organelles, like each one inside the cell?
No, that was actually more part of it. There may be some reference to it where I have told you that particular metabolic pathway or something is tied to an organelle, but you're not going to be asked about organelle structure or anything like that. Yeah.
But like
this test is on membranes and membrane function, and then the start of the metabolism part that we did, and that was all the way. Let me just see. Have I got the right one? Oh, sorry. Yes. So it was membranes, membrane function, and then this metabolism and enzymes.
Okay, thank you.
All right. I'm just flipping back to the beginning. So this is the start of this here is the start of membranes. I just added it to this slide set. So I tell you what I'll try and do. There's an awful lot of slides that we would have to go through. But what I'll start doing is I'll start flipping slowly from the start of membranes and membrane function. I'm just going to like give you the headline, okay, on each. See if this stimulates any questions along the way. All right. So membranes. Let me just do this. Sorry. All right. So membranes, membrane function. The first thing we did was the actual structure, molecular structure of the membrane itself. Remembering it's a bilayer. That bilayer arises because each of the phospholipid layers is made up of phospholipid molecules like this, one part of which is hydrophobic, the tail's there, the tail's there, and one part which is hydrophilic, the head's there, and the head's there. The only way that this, when we mix this in water, the only way it can satisfy all the requirements is if it forms this phospholipid bilayer like this. And that was the typical. That's only the skeleton, because the actual living nature of the membrane is conferred by all of the proteins which are embedded in the membrane. Those proteins have many, many different functions, many of them enzymatic. It's the proteins that actually confer the living nature to the membrane. This is called a fluid mosaic. And the reason for that is this is actually liquid. The phospholipid layers is liquid, and these proteins float in them. And remember that this is not nothing about this is random. Everything is carefully controlled by the cell. And one of the ways in which control is exerted, for example, is that the proteins are associated with cytoskeletal elements underneath. You can see them here, and they can be moved. The cell can actively move these proteins around inside the fluid phospholipid layer. Remember also that externally, there's all sorts of stuff which structures the cell, and which also in prokaryotes will allow the cell to be tied to its environment, everything else. So the membrane itself actually communicates with the outside, and it communicates with the inside as well. When we look at the structure of these proteins that are associated with the membrane, those which span the whole membrane are called the transmembrane proteins, or the integral proteins. This is a typical example here. And all of this area of the protein here, there, must be hydrophobic. It must have hydrophobic side chains associated with it so that it sits easily inside the hydrophobic area here. This area out here must be hydrophilic because it's associated with the hydrophilic heads and with the water, the aqueous medium there on either side. But remember also that there are peripheral proteins. There are peripheral proteins. Those are just associated with the outside of, with one side of the membrane. Most of the peripheral proteins are on the inside, in the cytoplasm. There are some peripheral proteins which are on the outside, but they're much more common on the inside here. When I say that the proteins confer living structure to the membrane, this is what I mean. They have a whole range of different properties. They transport in ways we've already discussed, but we can get to again. They have enzymatic activity. I've mentioned the respiratory enzymes, for example, in the prokaryotes are lodged in the cell membrane. They confer information from the outside to the inside to the cytoplasm. For example, there are proteins which are able to perceive specific molecules or specific in environmental conditions, pH, oxygen levels, these sorts of things, and confer that information, transfer that information to the cytoplasm. Take note, these are not transporting. This is not transporting the signaling molecule. It's binding the signaling molecule, and it's generating a signal, which it sends to some aspect of the cytoplasm. Proteins also are used for recognition. They label the outside of the cell, both for other cells to be able to interact with them usually. They may also act as anchors. They join cells together one-to-one, but they also join the proteins to elements, to the cytoskeletal elements. So they can bind, for example, to the external extracellular matrix, but they can also bind to internal elements of the cytoskeleton, and they can actually bind also to motor proteins, and the proteins themselves can be moved inside the membrane. So there are all these different functions for the membrane-associated proteins. Remember also, there's all sorts of proteins that are not membrane-associated, right? Not all proteins and enzymes are associated with the membrane. There are many which are associated with other functions inside the cytoplasm. Be sure that you understand the concept of permeability and of selective permeability. That is that the membranes are selectively permeable. They allow certain things to get across easily, and they stop other things from crossing. And the most important aspect of the membrane that does that is that hydrophobic layer, double layer inside. It is very, very difficult for most things, for most solutes, to cross that layer. That is very useful for the cell because it means that this thin layer of phospholipid is an extremely good barrier. It forms an extremely good boundary, and the cell exerts control over what comes across through its proteins, through the proteins which control the passage of material. So we spent some time talking about the nature of diffusion, especially across a selectively permeable membrane like this. And the one concept which I tried really hard to explain to people, what is really key to understanding the movement of water especially, is that you always remember that all substances, no matter what they are, whether it's gas, liquid, whether it's water, whether it's a solute, whatever, they tend to move down their concentration gradient from an area of high concentration to an area of low concentration. That's relatively easy to understand. What is sometimes difficult initially to conceive of is that water is a substance just like any other, and it also moves down its concentration gradient. So if I have a solution where I have a lot of solute, and in this example, what I've got here, I have a lot of, I think they said sugar here. Yeah, I've got a lot of sugar on this side of this U-bend, this U-tube here. When you think about it, initially we're going to start off, here's the volume here, it's the same as the volume here. But here, a lot of the volume is taken up with sugar molecules. Sugar molecules do two things. First of all, they themselves occupy space. The second thing is they actually, water molecules actually attach to them, like because of they are polar. And as a result, the amount of free water molecules on this side is much lower than it is on that side. The number of free water molecules in this same volume, the number of free water molecules is much higher this side than it is that side. Water is going to attempt to move in that direction here. It's going to move down its concentration gradient from high to low. But this is in the middle here is a selectively permeable membrane. The sugar may want to go that way as well, but it can't. The membrane stops it, but the membrane allows water to go. As a result, the water will carry on moving across this membrane until the water concentration on this side is equal to the water concentration on that side. That movement of water, the diffusion of water, has the special name osmosis. So this is osmotic movement. It's the diffusion of water itself. Okay. And that's an important concept. The cell, in actual fact, the cell membrane has special water passages called the aquaporins. They are proteins that specifically allow water to move across, where other things can't move across the membrane easily. Water moves across the cell membrane very easily through the aquaporin proteins. If we think about that, now you think about a cell. The inside of the cell actually has got a much higher concentration of solutes and all sorts of things. It has a much lower water concentration usually than the outside of the cell. So in fact, water continually attempts to pass into cells in general. Here are three different experimental situations. We're going to take a red blood corpuscle, which is just a cell membrane around the cell, as in any cell. We're going to put it into different situations. We're going to put it first into a hypotonic situation. That is a low concentration of solutes. In other words, a high water concentration. And the water, because of that, water is going to try to move into the cell continuously. The cell's going to swell, and eventually it's going to burst. It's a very dangerous situation. Fortunately, in our bodies, our red blood corpuscles actually swim in a fluid which is isotonic. That is, it has as much solute outside as it has inside. As a result, in an isotonic situation, it's not that water movement stops. It's that as much water as moves in will move out. Well, think about it this way. If I've got an isotonic situation, I've got the same water concentration here as I have inside the cell, and I let a water molecule go in, I've increased the water concentration inside, so one molecule will come out. That's what an isotonic situation is. As much moves one way, moves the other way, and the concentrations stay the same. If I take my red blood cell and I put it into a hypertonic situation, then the outside here has got a much higher concentration of salts. Water is going to flood out of the cell, and the cell is going to shrink. In both plants and in prokaryotes, this is also a very dangerous situation because the cell membrane pulls away. Remembering, plants and bacteria are the two things that have cell walls. The cell pulls away from the cell wall as the cell shrinks. And that's very dangerous. If it's not restored quickly, the cell will die. A reminder that animals, especially that are immersed in a hypotonic environment, they have to deal with this influx of water the whole time. Many of them have special mechanisms like water pumps that see to it that the water is pumped out as quickly as possible. In bacteria and plants, they allow the cell to swell until it swells up against the cell wall. And then the swelling stops, and as much water flows in, flows out. So that is how they regulate. I'll just remind you about this example, sort of an interesting thing, that the fish evolved in fresh water. So there they have quite a low tonicity to their body fluids. We automatically think that of fish as being seawater things, and that they must be the same, their body fluids must be the same as seawater. It's not true at all. In fact, they have quite a low solute concentration compared to seawater. So in fresh water, they have to face the same problem that all freshwater animals face. Their cells, because they are living things, they are full of solutes, they're full of molecules and things which are working and everything else. As a result, in freshwater fish, they have the problem that they effectively drown because water floods into their cells continuously through osmosis. And as a result, they have to work really hard to avoid getting flooded with water. They produce a very abundant quantities of a very, very dilute urine. And they have special mechanisms to recover ions, to conserve ions so that their solute concentration stays as high as possible. But remember, freshwater fish, in fact, drown if they don't have these mechanisms to pass water very, very quickly. In salt water, they have the opposite problem, because in fact, the salt water around them in the seawater has a much higher salt concentration than their body fluids. And both freshwater and saltwater fish, they have massive surface area that interacts with the water. This is one of the ways they respire. They respire through gills and also through the skin. So they have huge surface areas that water exchange takes place across. And in saltwater, in seawater fish, they continuously lose water. Fresh water floods out of them continuously, and they are a danger of dehydrating. Freshwater fish drown, saltwater fish actually dehydrate. And as a result, in saltwater, they drink continuously, and they actively excrete the ions, especially salt. They excrete ions through the gills and through the kidneys. And they try to conserve as much water as they possibly can. So understand the difference between the two and why those situations arise. This is the special situation. This applies to plants as well as to bacteria, as I've mentioned. Put them into a situation where their environment is hypertonic and they lose too much water, then they plasmolyze. The cell pulls away from the cell wall, as it does here. And that is plasmolysis. There's the word there. Okay, so the next really important set of concepts were the concepts about actual movement of ions by these different proteins across the membrane. Remember, this phospholipid membrane is a very good barrier. There's not much that makes its way across here. Gases will go across, water will go across very slowly, some ions will go across very, very slowly. But all of those processes take place too slowly for it to be biologically useful. Instead, control is exerted by proteins that are embedded in the membrane. And the first set of proteins are those that allow for diffusive processes to take place. So diffusion down a concentration gradient from high concentration of solutes to low concentration. And this is referred to as facilitated diffusion. Diffusion on its own would just be stuff going across the membrane. Facilitated diffusion is because there is a protein that allows it to take place. This is called a channel protein. I mentioned it already. Channel protein, they're very selective in what they allow across. They often just allow only one particular ion, one particular molecule to go across. And the movement is in the direction of the concentration gradient. No extra energy has to be put in. All you have to do is open this channel and the diffusion will take place. This is another kind of protein, is a carrier protein, and it's different in that the protein changes shape. It opens to the area that is high concentration, binds a molecule, and that binding causes the protein to change shape and pump it to the other side. But again, this does not require any additional energy. The energy is provided by the potential energy of this concentration gradient. However, many substances need to be moved into the cell against their concentration gradient. They need to be pushed into the cell from low concentration area to high, to an area that is already high in concentration. That is the case for many substances that the cell needs. And in order to get those across the membrane, the cell has to put in energy. This example shown here is only one of many such systems. But this is one that we always use because it's a classic system, and we will hear about it again when you talk about nerves and about how nerves function. And this is the sodium-potassium pump. Again, notice this is a carrier protein. Carrier proteins are those which change shape. But this is a carrier protein which changes shape in response to an input of energy and an interaction with the ions. So this is the inside of the cell. This is cytoplasm here. These proteins bind 3 molecules of sodium. There are the 3 sites there. When that occurs, it actually changes the nature of the protein, and it enables it to be phosphorylated. So this is ATP here. ATP is donating a phosphate group here and phosphorylating this. When that happens, the protein can actually change shape, and it opens to the outside, and the pumps out. Once it does that, the shape of these sites here changes, and they won't bind the sodium anymore. They pump it to the outside, where there's already a high concentration of sodium. There's also on the outside here, there is dissolved is potassium, and these sites here are potassium sites. Once it's open to the outside like this, it binds potassium. When it binds potassium like that, it loses its ability to hold on to the phosphate, and it dephosphorylates. When it dephosphorylates, it returns to its original shape. And it, at the same time, loses its ability to bind potassium. So it liberates the potassium to the inside. The take-home point from all of this is that this system is pumping ions, pumping sodium, and is pumping potassium to areas that are already high in concentration. And the only way it can do that is by having that protein be activated and energized by binding of phosphorus from ATP. And that drives the changes in shape that enable the protein to pump things one way or the other. Most proteins that are performing this active transport are going to rely upon ATP as a source of energy. So all of the proteins that are performing active transport are going to interact with a phosphorus source like this in order to gain their energy to do their pumping. Okay, this is what I set for you for extra credit. Many of you, by the way, have sent. And it was to explain the difference between these different kinds. So it should be familiar to you. Let's stop there for a moment. And here, are there specific questions on that section?
Do we only have to know a general idea of it, or like exactly how it works? Like,
the more detail you know, the better. That's
okay.
what I meant by studying in depth. The more detail you know, the better. Okay.
No, I know that. But
yeah,
I'm just
the reason, but there's an important reason for it, and that is the overall concepts don't sit easily if you don't see how they fit into the picture of how the cell functions. Otherwise, they become little factoids, and it's harder to understand them and remember them, even though it's more effort to learn the detailed structures.
Okay,
but
thank you.
As far as I'm concerned, I don't examine much beyond what you get in my presentations or my PowerPoint presentations, and in the the assignments that are set. So stay within those bounds and you'll be fine. You don't have to go and study all the detail that is in your book. There's far more than you can assimilate.
Okay, thank you.
Okay, the last thing to remember is just this system here, which is a system, it's called co-transport. And we pay particular attention to this because it becomes so important. Similar systems become very important when we talk about other metabolic processes such as respiration. And co-transport is the interaction of two distinct systems. The first system is one which is an active transport system. And in this example here, this is a proton pump. It is pumping protons to the outside of the membrane here. So when it does this, it can do this for many different reasons. The cell can use this in many different circumstances. But when we do this, what we're doing is we're building up what is referred to as a Kemi-osmotic differential. So first of all, there is a change in charge. The outside here is much more highly charged, positively charged than the inside. The inside is more negatively charged. The second thing is this is an actual chemical difference because we've got more protons on this side. The third thing is we've got a pH difference. The pH on this side is much lower than it is on that side. All of these things drive a potential energy difference, but across that membrane, a very powerful potential energy difference that is going to strive to be equalized if it possibly can. So what the cell, how the cell uses that is that it opens up some sort of a mechanism for these protons to flow this way. What they're doing is they're equalizing the charges on either side. They're equalizing the concentration gradients, and they're balancing out the pH. So they are moving down. We put in a lot of energy here to pump them to the outside. This is potential energy. When we open up a channel for them to flow this way, energy is actually released. That potential energy now can be harnessed by the cell. And it is harnessed by the cell to pump a substance along with the proton as it comes this way. In this case, what it's doing is it's taking sucrose and it's pumping sucrose into an area where there's already a high concentration of sucrose. Maybe this is a plant cell that is accumulating sucrose, for example. So it's still an active transport system, but it's just that a different protein here, the proton pump, is the one that actually expends the energy. After that, this is just harnessing potential energy that is built up from this system here. But this is an important one to pay attention to simply because this is a system which we're going to hear about. A similar system appears in many different guises in the functioning of the cell. Okay, so I won't spend much time on this. Just remember, please, the processes of exocytosis, pushing stuff to the outside, endocytosis, taking stuff in to the inside, and pinocytosis, little small sips, tiny vesicles that pull in material from the outside. These are all methods of bulk transport. Those diffusive methods we've talked about, that was for moving molecules. This is for moving actual substances, even things as big as foodstuffs, across into the cell, into and out of the cell. So we saw the three different kinds. Phagocytosis, that's taking in really big items like food into the cell. That's the sort of thing that our intestinal cells do, but it's also the sort of thing, for example, that paramecium or amoeba, those sorts of things do to feed. Pinocytosis is the formation of very small vesicles which sip solutes from the outside. And I'll give you one place where this is very, very important. That's in our immune system. We have cells in our immune system that continually sip the outside like this to test for the presence of pathogens that are floating around in our body fluids. There's one thing to explain. We don't do it in much detail, but I will tell you, just remind you, this is a coated pit. It's called a coated pit because the cell lays down these proteins against the membrane. It could do it anywhere. It could do it here. And these proteins have a tendency to crystallize as a sphere. And as they do that, they pull the cell membrane down with them to form this small vesicle. In receptor-mediated endocytosis, this is the other kind of endocytosis. Specialized proteins embedded in the membrane bind certain substances. And when they do so, they cause the coat proteins to be laid down. So they bind, they signal to the inside coat protein is laid down and it pulls a vesicle into shape like this to form another coated vesicle. And the tops of those proteins that were on the outside, they're now on the inside here of the. This can be done for many reasons, but one of the things that we may find is that this may be a way of binding hormones and transporting them to the inside of the cell where they can then have some activity. There are many other reasons why the cell would do that. It's just phagocytosis again, pinocytosis and receptor-mediated. Have a look at those diagrams. That's the summary. Okay, so before we move on, any specific questions about all of that? Okay, so we'll move on to our section on metabolism. And I just remind you about the two great grades of metabolism that we talked about. The first, the autotrophs. The autotrophs are those organisms which are able to take simple molecules, carbon dioxide and water, and bind them together with a source of energy to form a biomolecule such as glucose. So the vast majority of this kind of autotrophy that we see on our planet is done by organisms which use sunlight. So those are called the photoautotrophs, but there are a few organisms which are also able to do it with chemical energy. And those that we call the chemoautotrophs, and they're all prokaryote. The other great group of organisms are those which depend entirely upon biological molecules which have already been created. They've already been made by autotrophic organisms. That's us, for example. We depend entirely upon plant material or upon animal matter that has been derived from plant material. And we depend on them for both for a source of molecules to make our biomolecules, but also as a source of energy. So we're deriving our energy from things which have already been created. And those, we are therefore known as heterotrophs. And the next set of definitions that we need to have clear is that the overall, all of them, the reactions that take place that result in a cell being alive, organized and alive, moving material and energy around in a purposeful manner inside the cell. All of those reactions are referred to as metabolism. But metabolism is actually comprised of two parts. There are systems of breakdown, of catabolism. The ones that we pay the most attention to are those breakdown of fuel molecules to provide the cell with the raw materials to build things, but especially to provide the energy to do so. The reactions of building, of making molecules, making large molecules, are called the anabolic reactions. The breakdown reactions are the catabolic reactions. Catabolic reactions in general are exothermic. They release energy and the cell harnesses that energy. It doesn't just waste it all. It manages to get some of that energy and put it into a biologically useful form. The anabolic reactions require an input of energy. They are usually building molecules which would not normally arise at any appreciable rate. And as a result, we have to put in energy in order to see that those reactions go to completion. So remind yourselves, just read through this again, please. The nature of energy, and remember the different kinds of energy and the two laws of thermodynamics that we pay attention to. The first law, that the energy of the universe is constant. It can be transferred, it can be transformed, but it can't be created and it can't be destroyed. That means we're all dependent on a source of energy. Okay, plants depend primarily upon the sunlight as their primary source of energy. They take it and they transform it. They transform it into potential energy lodged in chemical bonds as chemical energy. We, in eating, we transform energy from one form to another. We eat something, we transform some of the energy into the energy of movement or whatever we need energy for. Energy can be transformed in the cell. But unfortunately, the second law of thermodynamics tells us every time we do that, every time we transform energy, we're going to waste some energy. Some amount of energy has got to be degraded to a lower form of energy to contribute to the entropy of the universe, to the disorder of the universe. We see that because we produce heat. The primary waste product of all of these reactions, energetic reactions, is that some of it is lost as heat, and heat is not biologically useful. We can't take heat and use it to build molecules, for example. So remember the bear, first law is transforming chemical energy into other forms of energy. Second law, here he is transforming chemical energy into the energy of movement. That transformation is going to release heat. Why? Because he's broken down molecules. He's broken down fuel molecules to their simple components, again, carbon dioxide and water. Use some energy, but he has to, unfortunately, pay his dues to the universe. And that is paid in the form of heat, which is ultimately radiated out to space and lost. So when we look at reactions taking place inside the cell, the first are catabolic reactions. Catabolic reactions are reactions which actually release energy. And in general, although they are enzymatically governed, once they are started, they proceed spontaneously and release energy, which the cell can actually make use of in one form or another. So we say the catabolic reactions in general are exergonic. They release energy, which can be harnessed by the cell. The cells of building, of making biomolecules, for example, taking simple components, taking monomers and joining them together. Even in plants, taking simple things and making sugars. These are all anabolic reactions, reactions of building, and they require an input of energy. So we have the products end up containing a much higher amount of potential energy than the reactants. In the exergonic reactions, the products end up containing less energy, and the difference in energy is harnessed. But here we have to put in energy. And if we look at any reaction, I'll just flip back, sorry. I'll just refer to this and then refer to it again. In any reaction, such as this, we're going to have to put in a lot of energy just to get the reaction to take place at all. And we need to think about how the cell now moves energy around. What is the form in which it extracts energy and moves energy? So in order to do so, what the cell does is it couples exergonic reactions with endergonic reactions. It's going to perform some exergonic reaction, which will release energy, and it's somehow going to grab that energy, and it's going to move it to the endergonic reactions that require an input of energy. The most common way the cell does this, most biologically useful carrier of energy, is this molecule here, which is ATP. So ATP is the energy carrier. And it carries energy in this bond here. This bond is actually highly energetic, quite difficult to make, but very, very unstable. So there are lengthy processes which the cell undergoes in order to make ATP. And ATP is a nucleotide. You can see it's got a sugar, ribose sugar. It's got a nitrogen-rich bases here. But here it has a triphosphate. And this is ATP. The substrate molecule for ATP is ADP. Here it is here. Sorry, here it is here with 2 phosphates. So this molecule here, ATP, lodged in here is energy. Where did that energy come from? It actually came from the electrons which were present in hydrogen molecules in some fuel source. That's where it originated. We are going to learn much later on. You haven't learned it yet. We're going to learn how that arose, how we transferred that energy from one set of chemical bonds into another set of chemical bonds. But that's its origin. So ATP is used as a carrier of energy, and it is a short-lived substance. It is manufactured as it's needed, and it's used almost immediately. Here it is here. When it is broken down, when it's catabolized, the triphosphate breaks here and leaves us with ADP, 1,2 phosphates and the adenosine there. There's adenosine diphosphate. Here's the phosphate group, but most importantly, here's the energy. And the energy, I told you before, I don't like that they put it on opposite sides. It should be together here, because often that energy is transferred together with the phosphate. You saw an example already in the transport proteins that were phosphorylated. When they were phosphorylated, what was important was that they were not only gaining the phosphate, they were gaining this energy from ATP. Okay, so here's an example of typical kind of metabolic reactions. Many of them are not spontaneous. Here's one with this is glutamic acid and ammonia. We can take glutamic acid and ammonia, mix them together. We can leave them forever, and there will a few of them every now and again might actually bang into one another sufficiently fast that they reorganize their bonds and everything and form this substance, glutamine. But under normal circumstances, this reaction takes place very, very, very slowly, useless to the cell. Instead, the cell needs to be able to speed that reaction along. And in order to do so, it's got to put in energy, and puts it in the form of ATP. So here's glutamic acid. What the cell does is it phosphorylates the glutamic acid. And it takes that phosphorylated glutamic acid together with the ammonia, and that then proceeds to a spontaneous reaction to produce the glutamine. In addition, of course, it produces ADP and phosphate. So that's the basis of this reaction. This is the basis of a phosphorylation reaction to energize a substrate. But the one thing I want to point out to you is this, in fact, itself does not just happen spontaneously. In fact, we would have enzymes looking after all of these reactions as they go along. But the impact of the enzyme is to phosphorylate glutamic acid rather, and then allow that to react with the ammonia to form glutamine. So what you're doing there is you're coupling a highly endogonic reaction, which is glutamine plus ammonia to form glutamic acid plus ammonia to form glutamine. That's endogonic. You're coupling it with a strongly exergonic reaction, which is the breakdown of ATP and the release of phosphate with its energy. We've seen this. This ATP is harnessed in any reaction which requires an input of energy usually uses ATP. I say usually because there are one or two other nucleotides which can be used, GTP, guanosine triphosphate can also sometimes be used, but the vast majority are ATP. Things like transport, movement, all of those sorts of things, they all use the same systems. So if we're continuously using ATP, we're going to have to make it somewhere. And ATP is manufactured through various means. We cycle ATP. We cycle ATP through the cell the whole time. So we'll take ADP and phosphate. We're going to put in energy into that system, and we're going to phosphorylate the ATP. This energy here that's coming in, that's going to be put in there, is derived from various sources. In plants, it's derived from sunlight. In heterotrophs, it's derived from food, from the breakdown of food. And the energy is used to manufacture ATP. The ATP is then allowed under controlled circumstances, particularly under the control of enzymes, is allowed to break down and drive endogonic processes such as the ones we've seen. We end up with ADP again, and we cycle it back again. So all of these systems require the use of enzymes. Okay, before I go any further, let's stop there. If anybody is still listening to me rabbiting on, if you have particular questions. Anyone?
The whole like thing of like ATP and like ADP, like I don't really understand it. Like if you could like re-explain that.
Like here.
Like no, the slide, like I think 2 slides to that. Yeah, that one.
Okay. All right. So I'll start. That's important. So I'm quite happy to repeat.
All right. Thank you.
So here's a reaction: glutamic acid plus ammonia to form glutamine. The cell does this the whole time. It needs to manufacture glutamine the whole time. Glutamine is an amino acid, and it's required the whole time by the cell. So the cell has to somehow or other drive this reaction. I can take glutamine and ammonia and mix them together in a vessel, and almost nothing's going to happen. The reaction is not going to take place spontaneously. It just in the chemical nature of the two is such that they're not going to react spontaneously with one another. Instead, this is a highly endogonic reaction. It's going to require an input of energy in order for it to happen. Now that might sound like it's a disadvantage, but it's not. It's an advantage to the cell because it means that this reaction is going to take place under highly controlled conditions. So we have this is how the cell actually puts in energy into this reaction. ATP is our most useful biological energy carrier, and the energy is contained in that very last phosphate bond of ATP. If I can break that last phosphate bond from ATP and instead bond the phosphate to another substance, what I'm actually doing is transferring energy contained in the electrons to the new substance, to the new product. So think of it this way. This is a set of chemical reactions. I've got glutamic acid and I've got ATP, and I'm going to react them together, and I'm going to end up with new products. So here's my substrate. Here is my product. My product is going to be phosphorylated glutamic acid intermediate. So this now has got a phosphate group attached to it, and we liberate ADP. Forget about it, off it goes. All right? Because what we're interested in is this phosphorylated intermediate. These phosphorylated intermediates are usually very unstable. They have a lot of potential energy locked into this phosphate bond here. And when I now put this together with the ammonia, the reaction takes place spontaneously. The that phosphate bond breaks and the glutamic acid reacts with the ammonia to form glutamine. So what I've done is I've taken a highly unlikely endogonic reaction, which requires an input of energy, and I've put in energy from this ATP. And as a result, now when I take this phosphorylated intermediate and put it together with ammonia, the reaction proceeds spontaneously, and I end up with glutamine. I've tied an exergonic reaction, the breakdown of ATP, to a highly endogonic reaction, glutamic acid and ammonia. And because I've put in the energy, I end up with my product there. And the only complication that I introduced was to tell you that this whole process, because it's our next topic, this whole process is guided by the presence of specific enzymes, which see to it that these reactions take place as they're supposed to do. Does that make it any clearer?
Yes, a lot. Thank you.
Okay. Okay, so let's just move on quickly to this discussion about enzymes themselves. Here's another example of a reaction which actually takes place really slowly. If I take sucrose, sucrose is a very stable molecule. I can take sucrose molecules and leave them. They will just sit there as sucrose for thousands of years. Every now and again, one of them might be struck by a cosmic ray or something and acquire enough kinetic energy that it vibrates enough to break its bonds. It is a disaccharide, and here's a bond here tying two sugar molecules together. Every now and again, one may break to form glucose and fructose. But though that reaction takes place so slowly, in biological systems, it's completely useless. The reason it takes place so slowly is that there's got to be an input of energy. There's got to be an input of energy. We've got to raise the kinetic energy of this molecule high enough that the bonds get weakened. In other words, we've somehow or other got to do something that's going to weaken those bonds and allow the bond to break, to give us our two monomers, glucose and fructose. What if the cell actually needs a source of energy and it needs to break sucrose, it needs to break down sucrose? Well, it will have to do it by using an enzyme system to do so. In the cell, we use an enzyme called sucrase, and sucrase very easily breaks this bond here. Very easily and very rapidly allows this reaction to proceed. You can always tell an enzyme by those last letters, ASE. As soon as you see ASE, know it's an enzyme, and this often tells you what it breaks down. Sucrase breaks down sucrose to glucose and fructose. The way in which enzymes work is that I described to you how a molecule of sucrose has to acquire enough energy that that bond is weakened and breaks. That we call the activation energy. And so for any reaction like that to take place, there has to be an inherent amount of energy associated with the reactants. In other words, have a look here. We've got this is reactant here, this AB compound, CD compound. These are relatively stable. If I want these components to interact with one another, I'm going to have to weaken these bonds here. Somehow or other, I've got to actually increase their kinetic energy. For example, the rate at which they are moving. I can do that with heat, perhaps. I can hit them with a cosmic ray. There are various things that can happen, but I have to get them moving. And I have to get them moving so that these bonds are weakened, and also so that when they smack into one another, they will shuffle the bonds. They will shuffle their electron, the distribution of their electrons, so that they form new bonds. And I might have to put in quite a considerable amount of energy in order for this to take place. The first thing is I've got to weaken them, get them moving so fast that these bonds are weakened. That is called the transition state. Once I reach put in a requisite amount of energy, then the reaction can take place spontaneously and they can shuffle everything around. Look how they've changed. Now we've shifted these bonds to here and here, AC and BD. Okay, we're not worrying about free energy and everything in this class. But the net take home is we have to put in energy here. This is the energy of activation. And for most biological reactions that take place in the cell, the energy of activation is very high. And the higher the energy of activation, the less likely it is that that reaction can take place spontaneously. In the case of sucrose, for example, you have to put in a lot of energy in order to get the molecule vibrating or wobbling so fast that it will break this bond here. Okay, so the cell makes use of an enzyme to catalyze the reaction, and the way in which enzymes work is they lower that energy of activation. So because they lower the energy of activation, often they lower the energy of activation tremendously. Because they lower the energy of activation, the rate at which the reaction takes place is much, much higher. That is the single most important property of enzymes. They lower the energy of activation. And because they lower the energy of activation, they speed up the rate of reactions. Reactions which would have taken a long time now take place very rapidly. And the enzymes themselves can be reused. They perform their activity, they spit out the product, and they can do the same thing over and over and over again very rapidly, so that they speed up the rate of biological reactions enormously. I've done this already, but that's okay. We can redo it for those who perhaps weren't listening. Most enzymes in the cell are proteins, not all. Remember that RNA also has enzymatic ability. But most enzymes are proteins, very complex proteins with a very complex three-dimensional shape. But they have one area of the enzyme which is able to perform that catalysis, to catalyze the reaction. And in order to, this is called the active site. And in order to do so, the active site binds the substrate. They are very, very specific for their substrates. Once they're bound, they form what is called an enzyme substrate complex. And very often when that happens, the shape of the enzyme will actually change. In binding the substrate, the enzyme can do various things to the substrates in order to foster a reaction. So for example, the enzyme may take two substrates, and in normal circumstances, we allow the substrates to mix, while they're bouncing all over the place. They hit into one another in the wrong orientation the whole time. They don't hit with much energy, so they never react. The enzyme grabs the substrates and it puts them together in exactly the right orientation so that the reaction can take place. That's one way it works. The second thing, in case of sucrose, for example, the sucrase enzyme grabs the sucrose, and then once this enzyme substrate complex forms, the enzyme actually begins to strain that bond. It changes shape and begins to stress the bond between the two parts of the sucrose. The third thing is that within this pocket of the active site, the enzyme may create a highly specialized environment, depending on the enzyme, depending on what reaction needs to take place. You may have within here a highly hydrophobic environment, for example, or a very, very acid environment, or something like that, something that fosters the reaction. So that's the third. And the fourth thing is that the enzyme may temporarily covalently bond to the one or more of the substrates, changing their bond orientation, but also may temporarily add things like a phosphate group to the substrates. So the enzyme itself can interact in those energetic processes that we talked about before. So all of these things make it much more likely that the correct reaction will take place and that it will take place quickly. As soon as it does so, the enzyme loses its ability to bind the products. Its affinity is for the substrates. It loses its affinity for them and it spits out the product. And then the active site is there, ready to interact with more substrate. So this is, go through this, and it gives you the summary of the. Are we all right with that so far? We're just going to go to 5:30. So if you have questions, shout them out, all right? Remember that because the enzyme is a protein, a lot of its three-dimensional structure is affected by things like pH, temperature, all of these things. So every enzyme has an optimal environment in which it works, and it's optimal because that is the environment where its shape is absolutely perfect and the shape of the active site is perfect. If we start fiddling with the environment, we affect the actual bonds in the protein. We're going to affect the shape of the enzyme and decrease its ability to work. So, for example, one of the really important things is the temperature at which they work. Most of our enzymes, for example, operate optimally at 37 degrees Celsius. Not unsurprisingly, that is our body temperature. We keep a constant body temperature for that very reason. All of our enzymes are constantly operating at their optimum. But there are organisms which have very different temperature optima. There are bacteria which have optima around 77 degrees Celsius. There are other that have optima around 90 degrees Celsius. It just depends on the organism, how they've evolved, and how their enzymes have evolved. Same for pH. pH is very important, especially because it affects the ability of the protein to form hydrogen bonds. Hydrogen bonds are what determine the three-dimensional structure in space, very largely. And as a result, if I start fiddling with the pH, I start affecting the enzyme's ability to form those hydrogen bonds, and the protein shape may begin to change. As a result, enzymes have an optimum pH as well. So this, I gave the example in us. Here's optimum pH for one of our stomach enzymes, around about 2, very, very low, because of hydrochloric acid in our stomach. In our intestines, it's just above neutral at 8. And that's the optimal pH for another enzyme called trypsin. So different enzymes in the same organism, radically different optimal pHs. And quite a lot of this I have already discussed. If you came in late to the class, I will post a recording of what we discussed earlier. Okay, I'll just mention again. The enzymes very often consist of different parts, and the protein part of the enzyme is called the apoenzyme. But often they require other substances to come in, bind with them in order for them to work. And those are called the cofactors. They are non-protein substances associated. Some cofactors are simple things like metal ions, that sort of thing. But some of them are actually quite complex organic molecules. And those we call the coenzymes. And those are things like the vitamins. Most of our vitamins are coenzymes that function together with enzymes. All right. I'm going to finish off by talking about inhibition systems. Just to remind you, we talked in class about two ways of inhibiting the activity of enzymes. The first is called competitive inhibition. This is not a way that the cell uses to control its own enzymes. This is a way to poison a cell. This is a way to poison an enzyme from outside and stop it from working. Here's our normal enzyme. Here's its active site. And I told you the substrate binds in a very specific position to the active site. A competitive inhibitor comes in and it occupies a part of that active site, and it usually does so irreversibly. It binds permanently, and it blocks the active site. It blocks it completely, and the substrates cannot bind there. This is a way of poisoning the enzyme. And a typical example is hydrogen cyanide, for example. Hydrogen cyanide poisons one of the respiratory enzymes in our cells, in our mitochondria. And that's why it's a poison. Other things, snake venom, spider venom, or these sort of things often are competitive inhibitors on enzymes. A cell controls its enzymes through a different system. And this is through non-competitive inhibition. Non-competitive because it is not competing for the active site. Instead, in non-competitive inhibition, there is another site on the enzyme where a substance can bind. And when it binds, it actually alters the shape of the active site temporarily. This is not a permanent binding, it's a temporary binding. It alters the shape of the active site, and the enzyme can no longer perform its catalysis. This is non-competitive, and it's extremely useful to the cell because most of our enzyme systems work in these metabolic pathways. Here is a metabolic pathway, taking threonine, putting it through a series of reactions to form isoleucine. This, however, is an enzyme which can be non-competitively controlled. We also call that allosterism, and this is an allosteric site. So if I bind a substance there, I'm going to stop this reaction from taking place. I'm going to stop these reactions. Typically, for an allosteric system, it is the first enzyme in a pathway that is allosteric. And the substance that binds at the allosteric site is the end product. So here's the same enzyme. Now it's binding the end product at its allosteric site. The active site is no longer available, cannot operate. And meantime, the cell is using up the isoleucine that has been produced. This is a temporary binding. It goes in, out, in and out, in and out. The lower the concentration of isoleucine, the less often this is going to be bound. The less often this is going to be bound, the more often the active site is going to be available. This gives the cell exquisite control because the higher the concentration of isoleucine, the product, the slower the pathway is going to run. The lower the concentration of isoleucine, the faster the pathway is going to run. And the last thing I'll say about allosterism is that there are systems also where the enzyme can be turned on. This is a system called feedback inhibition because the product is feeding back to the enzyme, to the first enzyme to stop it working. But you can also have a feed forward system where an enzyme is switched on by the presence of the substrate. And in that case, the substrate would bind here and activate the enzyme so that it can perform some other catalysis, not the system. This is a feedback, but remember that feed-forward reactions also occur. And I think for the moment that is all that I'm going to say. I don't think I didn't discuss, I may have discussed that. Yeah, I did just say, remember that enzymes are placed where they are most likely to be used. Respiratory enzymes in the mitochondrion, for example. Okay, so as we finish, are there any specific questions about this? I'll give you a second to think before I shut you down. All right, if there are no further questions, your test tomorrow consists of 40 multiple choice questions for 80 points and then 20 points written section, okay? And if you've gone through these sections that I covered today carefully, you should have no problems at all. All right. If there's nothing else, then I'll bid you good night.
All
And I'll
right,
see you
thank you,
tomorrow morning.
Professor, so much.
Thank
That's a pleasure.
you.
Sure.
Bye, have a great day.
Bye.